So that's antibodies and natural killer T cells. But how can we bring these two components together so that it can actually form a viable drug in the treatment of cancer? So this problem has been assessed, and the way of using antibodies to sort of force an immune response from the natural killer T cells is we engineer a new type of antibody, which is unlike the natural state of an antibody. We use the antigen presenting site on the light chain of the antibody for CD3, which is a particular component on the natural killer T cell. CD3, CD stands for cluster of differentiation 3, and CD3 are found on most natural killer T cells. Then instead of that antigen presenting site being attached to the larger component of the antibody, we attach that to another antibody which is targeted to CD19. CD19 is a protein found on most acute lymphoblastic leukemia cells. And so in this respect, we've almost got like a double-edged antibody, which has the ability to bind specifically to T cells and also specifically to acute lymphoblastic leukemia cells, which brings them together. Bringing them together results in this cytolytic synapse, which then forces the T cells to start an immune response against the malignant ALL cells. And that's essentially how BiTE technology works. There will be variations within the different target antigen presenting sites, but essentially that is BiTE technology, and this illustrated here with blinatumomab. The indications lifted directly from NICE. Blinatumomab is used in relapsed or refractory Philadelphia negative acute lymphoblastic leukemia, obviously expressing CD19. In practice, we use this to induce a remission for patients who have relapsed or have become refractory to the treatment for their acute lymphoblastic leukemia. And the idea is that we can salvage their remission or induce a remission, which will make them eligible for a hematopoietic stem cell transplantation. And in this group of patients, this will be an allogeneic stem cell transplantation, not an autologous stem cell transplantation. Lifted straight from the company's drug chart, the company's website. The way that blinatumomab works is a 3-step process. Step 1, the two components are targeted. Step 2, they engage, and at this point, the T cells and the B cells target B cells, malignant B cells, are brought to close proximity with the blinatumomab, and this starts the toxic delivery of these toxic enzymes. These toxic enzymes are inserted into the malignant B cell. This starts an inflammatory release of inflammatory cytokines and results in lysis of the target cell. And so in this respect, I think blinatumomab is a lot like Tinder, in that previously the natural killer C cells have swiped left to when they've encountered any B cells, but blinatumomab forces the natural killer T cells to swipe right. Why target CD19? Well, CD19 is a very good target in the fact that it's expressed in most ALL cells, and in fact, only 10% of ALL cells do not express CD19. So it's a good target in that respect, and so you've got quite a wide range of good percentage chance of having a CD19 positive ALL. To illustrate that, if you were working at the Marsden and you wanted to see if your patient was CD19 positive or not, look in lab specialist pathology, and it's the immunophenotyping that will actually show you show you if the patient is expressing that particular protein. And this is a patient who is. The efficacy, the medium overall survival in a trial versus FLAG-IDA showed a positive survival outcome. Survival of 7.7 months in the blinatumomab arm versus 4 months in the chemotherapy arm. Remission rates within 12 weeks after treatment were significantly higher in the blinatumomab arm versus the chemotherapy arm. However, I wouldn't breathe too much credence into this for two reasons. The first being that within this trial, these patient groups were actually heavily pretreated, and obviously blinatumomab requires functioning natural killer T cells, and after patients who have received multiple lines of treatment, their marrow becomes less effective, so they're less likely to have functioning T cells. And so if we're using blinatumomab more upfront, which we essentially will be doing for patients who've only gone through one prolonged course of chemotherapy, hopefully we will see better remission rates and better overall survival rates. And secondly, we're not actually using blinatumomab to improve overall survival. We're using blinatumomab to induce a remission so that then we can give a patient a stem cell transplantation. And it's the stem cell transplantation that will give the patient hope of a long-term cure. Although saying that, there have been some papers suggesting that actually blinatumomab alone may be sufficient in some group of patients to actually induce a complete remission in its own, a complete long-term durable remission. However, that data is ongoing. Blinatumomab does cause side effects, and I wanted to draw everyone's attention to its side effect profile, which is kind of distinct and unique versus chemotherapy. Now, blinatumomab itself does cause serious grade 3 adverse effects, probably a little bit less than traditional chemotherapy, but still relatively high. Your chances of encountering some serious grade 3 or above adverse effect is 87% versus 92%. But the side effects are rather different. You're less likely in the blinatumomab arm to encounter myelosuppression, things like neutropenia and ultimately infections. However, you're much more likely to see a newer side effect to most of us, the cytokine release syndrome. Whereas in the other arm, the chemotherapy arm, cytokine release syndrome wasn't noted at all. Cognitive dysfunction has been well established with blinatumomab, but interestingly, it wasn't that dissimilar figures reported from the chemotherapy arm versus the blinatumomab arm. So, however, we'll discuss the neurological effects of blinatumomab in a second. So, the most serious side effect that is blinatumomab related has been defined as the most serious adverse effect of blinatumomab, which has been discovered, is the cytokine release syndrome or cytokine storm. Now, cytokine release syndrome is relatively self-explanatory. It's a release of large amounts of cytokines. Cytokines are a range of inflammatory mediators, and they include interferons, interleukins, chemokines, colony stimulating factors, and tumor necrosis factors. And most of the serious adverse effects that we get from cytokine release syndrome are from the interleukins. Cytokine release syndrome is a syndrome, so therefore has a collection of includes just a collection of adverse effects, making it difficult to diagnose. So clinical features of cytokine release syndrome are high fever, malaise, fatigue, myalgia, pain in the muscles, nausea, anorexia, tachycardia, cardiac dysfunction,